Welcome to all of you in today's session. Here we are discussing enzymology. Particularly today we are going to discuss enzyme catalysis and its mechanism, and then various factors affecting the activity of different enzymes. So this topic is basically very interesting, that how enzyme catalyze a particular reaction. Now we see that as I mentioned earlier, that enzyme catalysis is very vital for the functioning of living organisms. In conditions, relevant reactions that occur without enzymatic assistance are generally very slow. This is because most biological molecules are stable within the neutral pH, mild temperature, aqueous environment existing inside the cell. So basically many chemical processes common in non-biological settings are either unfavorable or unlikely within the cellular environments. So these include the transient formation of an unstable charge intermediates, or the collision of two or more molecules in the precise orientation required for the particular reactions. So enzyme catalysis is basically the enhancement in the rate of chemical reaction to the active site of a protein. The mechanism of enzyme catalysis is similar in principle to other forms of chemical catalysis. Basically, enzymes create a specialized environment that enables a specific reaction to occur more quickly. This process basically occurs within a confined area of the enzyme known as active site. Here you see active site, where substrate generally binds. The molecule acted upon by the enzyme is then bound, and the active site's surface is composed of basically amino acid residues with groups that interact with the substrate, facilitating its chemical conversion. The formation of the enzyme substrate complex, initially proposed by Charles Adolph Woods in 1880, it was just a breakthrough research and contributed a lot that how enzyme catalyze a particular reaction. Now, how basically enzymes work? Question arises that how enzymes work basically. You see that enzyme combined with substrate to form enzyme substrate complex. Then this complex is activated when this enzyme substrate complex is formed, and then enzyme substrate complex is dissociated. Enzyme is dissociated to form a product. So basically, as a catalyst, it serves to enhance the rate of reaction without influencing the equilibrium state. So this is very crucial to note, and this principle also applies to, of course, on enzymes, as you see that how enzymes bind, how the substrate binds on the enzymes present, and then it forms enzyme substrate complex, and then enzyme it is converted into product and enzyme is released. So basically enzyme facilitate the conversion of substrate to product and also capable to catalyze the reverse process, that is product to substrate. So the primary function of enzyme is to accelerate the transformation between substrate and product. Basically enzymes are not basically used in this process, leaving the equilibrium point unaffected. However, you see that the presence of appropriate enzyme significantly accelerate the attainment of equilibrium by boosting the overall reaction rate. So the majority of chemical reactions generally require the presence of enzyme catalyst, and typically under extreme conditions such as high temperature or varying pH levels, or they may require specific organic solvents. However, you see that enzymes facilitate these reactions under normal cellular conditions such as temperature of about 37 degrees C, a biological pH range of about 6.5 to 7.5, and generally it occurs in aqueous environment. And generally enzymes exhibit high efficiency in catalyzing the reactions, accelerating them to by factor ranging from 10 to power 6 to 10 to power 14 times faster than compared to the reactions without enzyme catalysis. Among the most potent enzymes is carbonic anhydrase with a turnover rate of basically 6.6 million 0.6 million per second, or 6 lakhs you can say. Even in, you see that is spontaneous thermodynamically feasible reactions where free energy of the product is less than that of reactant. Generally, if standard free energy change, that is delta G of the reaction, is negative, then the reaction does not start by itself. So therefore, a substrate must first come into the transition state, into the intermediate state, from transition state to into intermediate state before converting into the product. And the intermediate state is known as transition state. Basically, the energy that requires molecular rearrangement in a substrate molecule, which makes it easy for the substrate to get converted to product, and the free energy of transition state is generally higher than either substrate or product. So the initial point for both forward and reverse reaction is referred to as ground state. You see that here reactant is converted into product, and that how you see that this shows that a reaction catalyzed by enzyme and a reaction occurring without involvement of enzyme. So the basically this difference in free energy between the ground state and the transition state is termed as activation energy. This energy, you see that this activation energy differs in both the type of reactions. One is enzyme catalyzed reaction, another one is non-enzyme catalyzed reaction. So during the conversion of substrate to product, the change in free energy is basically plotted against the progress of reaction in a reaction coordinate diagram, that has been shown here. On one axis progress of reaction has been shown, and on another axis free energy change has been shown. So basically the substrate exists in the transition state for a very brief period, typically about 10 power minus 14 to 10 power minus 13 seconds before it is converted into product. Therefore, activation energy denotes the energy required, or you can say necessary, to initiate a reaction and acts as a barrier to chemical reactions. So basically a higher activation energy corresponds to a slower reaction rate. Here it is very important to note that the enzyme do not alter the equilibrium constant, but accelerate the reaction rates by reducing the activation energy. You see that here activation energy is more in non-enzyme catalyzed reaction, and you see here this is enzyme catalyzed reaction. And in both the course of reaction, you see that reactant is converted into product, but the activation energy is less in enzyme catalyzed reaction. But one thing is very interesting to note here, that standard free energy change is that is not change at all. So there is no change in the standard free energy of the enzyme catalyzed reaction. So in each step in reaction pathway involves basically interconversion of two sequential reaction intermediates. If there are multiple reaction steps in a pathway, the one with the generally highest activation energy becomes the rate limiting step. This is also a very important point to note at this juncture. Basically, these transition states occur when the enzyme binds with substrate to form enzyme substrate complex. As I mentioned earlier, the active site of an enzyme molecule is the site where it binds with the substrate. It is basically a three-dimensional structure formed by the folding of the constituent polypeptides, ensuring a specific conformation of the enzyme. While the active site, you see that occupies only a small portion of the enzyme's structure, but it is very crucial for the maintaining proper positioning of the interacting groups to prevent its collapse. And the residues within the active sites are responsible for both substrate binding and catalysis. In some enzymes, these residues may be the same. So, you see that in both the path, one is course of reaction without enzyme, and this is the course of reaction with enzyme. The activation energy is different, but you see here standard free energy change is unaffected by enzyme. So, it could also be exemplified with this that you see that this is the mountain existing here, and if we want to cross from one side to another side, it's very tough to cross. But if there is a existence of a tunnel, obviously it is very easy to go through a tunnel rather than climbing to a hill to reach the destination. So, enzyme, you can compare the function of enzyme is going through the tunnel, and without enzyme just crossing the hill. So, you see that here activation energy is less and here activation energy is more. So, you should just try to grasp the thing that how enzyme catalyzed reactions lowers basically activation energy. Now, try to understand how enzyme catalysis occurs. Basically the binding between the substrate and the enzyme's active site involves non-covalent bonds such as ionic, hydrogen, hydrophobic bonds, and various van der Waals interactions. The binding energy resulting from these interactions is basically responsible for lowering the activation energy. Just I mentioned that basically enzymes lowers what? This activation energy. So, in this regard, two models have been proposed to describe this binding process. One is lock and key model, is a basically a model which explains the enzyme substrate interactions, and this is proposed by Emil Fischer in 1894. So, according to this model, that enzyme acts as a highly specific three-dimensional protein configurations with unique active sites that selectively bind to a specific substrate, much like key fitting into a lock. You see, key fitting into a lock. The model basically suggests that once the enzyme and substrate bind, the enzyme catalyzes the chemical reactions leading to the production of product. Here you see that products are formed. So, basically the lock and key model does not fully explain for the stabilization of the transition state during the reaction, or it address the challenges changes in the enzyme conformation once the substrate bind. It's basically have certain limitations. So, this further leads to the proposal of another model that is known as induced fit model. So, basically this induced fit model was given by de Koechlin in 1958, which describes that formation of enzyme substrate complex or interaction of enzyme substrate, how they interact with each other. And according to this model, what the active site of enzyme is not very rigid, but it's flexible and can have different conformations. So, this model is a little bit different from lock and key model, that basically you see that binding of the substrate to the active site of the enzyme is not very much rigid, and it's flexible. So, and can have different conformations, So, the amino acids basically present at the active site determines the specific binding of the substrate with particular enzyme. Once the substrate binds to the enzyme, the conformation of the active sites gradually changes and attains a shape that is very much complementary to the substrate. So, this change in conformation is generally induced by the binding of the substrate and leads to the formation of enzyme substrate complex that is more stable and catalytically active. So, basically the induced fit model explains how enzymes can stabilize the transition state during the reaction and how changes in enzyme conformation upon binding of the substrate can lead to enzyme catalysis. So, this model was supported by experimental evidence and now widely accepted as a more refined view of enzyme substrate interaction than the previous model just I discussed, lock and key model. Now, type of catalytic reactions I will discuss that enzyme typically utilize multiple strategies to catalyze specific reactions, including a well studied mechanism like general acid base catalysis, metal ion catalysis and covalent catalysis. So, it is very common for enzymes to employ a combination of these catalytic strategies just I mentioned. Like for example, chymotrypsin provides a very interesting example where both covalent catalysis and general acid base catalysis are utilized. Now, what is acid base catalysis? The question arises, what is acid base catalysis? Basically, acid base catalysis involves the transfer of protons between an enzyme and substrate and can be categorized into a specific and general acid base catalysis. In a specific acid base catalysis, generally hydronium ions and hydroxide ions directly serve as the acid and base groups respectively. In contrast, general acid base catalysis involves the donation and acceptance of protons by other groups besides hydronium ion and hydroxide ion. In this case, generally ionizable functional groups present in the amino acid side chains and cofactors play a very crucial role in the catalysis by acting as acids and bases. So, for example, imidazole, hydroxyl, carbonyl, sulfonyl, amino and phenolic groups present in the amino acids serve as a very interesting example. But as far as enzyme catalyzed reactions are concerned, only general acid base catalysis occurs. An example of general acid base catalysis is the hydrolysis of peptidoglycan catalyzed by lysozyme. And a specific acid base catalysis is generally basically determined by pH and the buffer concentration and pH, and generally buffer concentration does not impact it. However, in general acid base catalysis, buffers may donate or accept protons in the transition state, thus could affect the rate of reaction. Now, what is covalent catalysis? Generally, covalent catalysis is a mechanism used by enzymes to catalyze reactions by forming a transient covalent bond between a substrate and residue in the enzyme active site or within cofactor. Basically, this covalent catalysis involves the formation of covalent intermediate that helps to reduce the energy of transition state in the later stages of the reaction. Basically, those enzymes employ covalent catalysis that possess a nucleophilic or electrophilic centers and which facilitate the covalent and nucleophilic catalysis by attacking the electrophilic centers of the substrate, forming covalent intermediates. Typically, you see that electrophilic centers in the substrate include phosphoryl group, acyl groups and glycosyl groups. And once reaction is complete, the enzyme reverts to its original position and then come in the unaltered state. And generally, this proteolytic cleavage of proteins by chymotrypsin is a very interesting example of this type. Generally, covalent electrophilic catalysis is also observed and involves coenzyme products that generally generate electrophilic centers, and Now metal ion catalysis is another type of catalysis that require metal ion for mm, catalytic activity. And there are two types of metal dependent enzymes. Metallo enzymes which generally bind the metal ion tightly, require it or to maintain its stable native state, and metal activated enzymes which bind generally metal ions weakly during the catalysis. So metal ion basically can function catalytically in several ways, including acting as electrophilic catalysts to stabilize the increased electron density or negative charge on the reaction intermediate that develop during the reaction. So these are the very basic type of catalytic mechanisms I had discussed. Now various factors affect the activity of enzyme. First is you see that activity of enzyme is generally influenced by various environmental conditions, just I mentioned temperature, and generally change in these conditions could alter the rate of reaction catalyzed by the enzyme. So several factors affect the rate of enzymatic reactions, including temperature, pH, enzyme concentration, substrate concentration, and the presence of any inhibitors or activator. So first you see that enzyme activity is generally affected by temperature, and with increasing temperature you see that the kinetic energy of the molecules increases, leading to generally more random collisions between molecules per unit time, including the collision between enzymes and substrate molecules. So this results in an increase in the rate of reaction and the formation of more product. However, what happens as the temperature goes on increasing or further increase in temperature, more bonds generally will break, especially weaker ionic bonds as a result of the strain, causing the rate of reaction to decrease. Therefore, the rate of enzyme activity basically increases with the temperature until the optimum temperature is generally reached, after which it falls, it generally protein start to denature. You see that in this uh, slide it has been shown, the graph has been plotted between the different temperature range and then activity of enzyme. You see that with the increase in temperature the enzyme activity increases, but after reaching a threshold level, that is up to 40 degrees C, the enzyme activity start decreasing. And the optimum temperature for most enzyme in the human body is around 37 degrees C. And you see after 40 degrees C the enzyme activity start decreasing due to the denaturation of proteins. Now second is the pH. Each enzyme generally you see that operates within a specific pH range that optimizes its function. So basically the presence of hydrogen and OH ions generally can disrupt the hydrogen and ionic bonds that maintain the structure of enzymes by being attracted or repelled by the charge within these bonds. This interference leads to alterations in the shape of enzyme, particularly its active site. Different enzymes have varying optimum pH values where the influence of H and OH ions on their internal bonds results in the most complementary shape of the active site to the substrate. At this optimum pH, you see that the reaction rate is maximum. You see that reaction rate is maximum, and then you see that extreme pH levels can disrupt the bonds maintaining the tertiary structure, including the active site, leading to generally irreversible denaturation of enzyme. So any pH deviation from the optimum generally swiftly reduces the reaction rates, as more molecules will have active sites with shapes less complementary to the substrate. Some enzymes like trypsin are active in alkaline medium, that is high pH, and pepsin basically shows optimum activity in acidic medium, while diastase in neutral medium. So basically the intention here is to show that each and every enzymes are active at a particular pH. Now third is the substrate concentration. So enzyme activity basically also depends on the concentration of substrate. Initially when there is an increase in the concentration of substrate, you see an increase in the activity of enzyme is generally observed. Once all the active sites of the enzymes are occupied by the substrate molecules, what happens? This phenomena occurs basically as high concentration of substrate leads to more frequent collisions between substrate and enzyme molecules, thus increasing the production of products. However, once a certain concentration, or you can say threshold, is reached, further increase in the substrate concentration generally ceases to influence the reaction rate. So at this point the substrate concentration becomes not a limiting factor, and generally saturation is reached. You see that further increase in the substrate concentration is not going to increase the rate of reaction. So this is the threshold level. Now factor next is concentration of enzyme. So generally small enzymes in very small amount is required for catalyzing a particular reaction. However, you see that the increasing the concentration of enzyme leads to a linear increase in the rate of reaction, provided that there is enough concentration of substrate, as more enzymes are available to collide with the substrate molecules. However, this effect is limited up to, you can say, a certain extent, certain concentration beyond which the enzyme concentration is no longer a limiting factor. So at this point basically adding more enzymes will not increase the rate of reaction anymore, and generally the rate of reaction increases with an increase in enzyme concentration or substrate concentration until a limiting rate is generally reached, after which further increase in concentration produces no significant change in the reaction rate. Another factor is water. Enzyme activity is basically significantly hindered when water is lacking, and generally in dry states most of the enzyme becomes almost in inactive state. So adequate cell hydration is very crucial for enzyme function, because water serves as both the medium for enzyme reaction, and in many cases it is one of the reactants. So thank you very much for patiently listening and the different crucial aspects of enzyme. Thank you very much.